Hello, everyone, and welcome to this week's EV Journal Club. We have a couple of questions in front of us today that are going to be addressed in this paper. I think one of them is one that we think about quite a bit, which is how do we define extracellular vesicles? And the other one is how do we make enough extracellular vesicles to do what we want to do, maybe therapeutically? So this journal club is going to be devoted to extracellular vesicle mimetics. So what is a mimetic and how do we use it? And today we're going to have the discussion led by Sara Elgamal of Ohio State University, who has selected this paper and prepared a nice presentation for us. So thank you so much, Sara, for stepping in this week and for sharing your time and your expertise with us. And I will hand it over to you now.
So thank you so much for letting me participate in this journal club. I'm Sara Elgamal. I'm a postdoctoral scholar at the Experimental Hematology Laboratory at the Comprehensive Cancer Center, Ohio State University. Next slide, please. So the paper I'm presenting to you today is titled Novel Alternatives to Extracellular Vesicle-Based Immune Therapy, Exosome Mimetics Derived from Natural Killer Cells. So this paper was done at the School of Medicine, Hongpu National University, Jeonju, South Korea. And just before I go into my slides, I just wanted to let you all know that I've been using the term exosomes extracellular vesicles according to the source that I used to prepare the slides. But of course, it's more accurate to use the term extracellular vesicles with all the knowledge we know now. Next slide, please. So a brief introduction about exosomes as drug delivery systems. So exosomes are endogenous vesicles from 50 to 150 nanometers in size. They're produced by almost all mammalian cells through the endosomal system. Exosomes, of course, can transport biological cargo like RNAs and proteins, and that's why they're very interesting to us. The advantages of exosomes as DDSs include their biocompatibility, reduced clearance rates, low systemic toxicity, facilitated cellular uptake as compared to several other synthetic DDSs. Also, exosome uptake and intracellular routing are a result of the presence of key proteins on the exosomal surface like tetraspanins and integrins. So that was correct. So we can, yes, please. Next slide, thank you. So challenges of exosomes as DDSs are that, of course, they require isolation through time-consuming and tedious techniques. We also get a low production of naturally secreted EVs. We get a preparation that is heterogeneous. We have to deal with cargo sorting and therapeutic loading. Thank you. So what are mimetics? So these are bio-inspired drug delivery systems. So there's several, you'll find them referred to as different terminologies in different literature. So there's my preferred terminology is cell-derived nanovesicles, so short for CDNs. There's also cell-derived mimetic nanovesicles, short for NVs, and then there's exosome mimetics, like what the paper that I'm presenting today uses. So you obtain these by subjecting cells to physical extrusion, thus producing, so that you produce the nanovesicles. You can achieve this by passing cells through an extruder, which is like a device where you would press the cells through it with membranes, or using microchannels, or using custom-made devices designed for centrifuges. So loading the therapeutics can be done by electroporation, or during the process of cell extrusion, or utilizing genetically engineered cells. Thank you. So I'm just showing you here a scheme from this from a paper over here where they used these devices that were designed for centrifuges. So you can see the spin cups and that are fitted with filters. And then you see the process here where you pass them by several filters and end up with the particles that are a specific size. And of course, there's specific procedures in order to separate these size from all the debris that are generated during this process. Next slide, please. The benefits of CDNs are that they generate larger yields, and it takes less time and is more cost effective. It does not require EV depleted culture media. They are potential alternatives for therapeutics which are not naturally sorted into exosomes. And then we could also get cargo loaded into the exosomes by we can get cargo loaded into these nanovesicles by actually starting with cells that are engineered to overexpress the cytosolic or membrane protein of interest. Next, please, thank you. So critical considerations that we need to keep in mind when we're talking about CDNs is that they combine the characteristics of cells and AVs. They vary from exosomes or EVs in terms of loading methods and surface functionalization. They differ in surface proteins and lipid composition. Many tools that we actually use for exosome research might not be applicable directly to CDNs because of the difference in the membrane properties and topology. Next, please. So back to the paper that I'm presenting today, novel alternatives to extracellular vesicle-based immunotherapy, exosome mimetics derived from natural killer cells. Next slide, please. Thank you. So just a brief background about natural killer cells. NK cells are innate immune lymphocytes that can destroy, infecting, or transform cells. NK cells are like, there are trials to use them in immune therapy, but they require prior to that you would take them ex vivo, expand them to obtain a sufficient cell number that would express activation markers and the necessary receptors, cytokines, enzymes, and preferrin. An interesting alternative or other promising aspect of NK cells is using NK exosomes that also express cytotoxic proteins and have anti-tumor activity. So due to the low yield and difficult and expensive purification, of course, of NK exosomes like any other type of exosome, this work was investigating how to generate CDNs from NK cells. Next slide, please. So before I go into their first figure, I just want to put out there a detail of the preparation method, just because we are all very interested in those kind of details. So if you look on the right side, that's how they prepare the NK exosomes that they refer to in all the figures as NK-EXO. And then if you look on the left side, you see the NK exosome mimetics, which they refer to as NKEM in this paper. In all the figures. So looking at the exosomes, they culture them, of course, in exosome in stem cell growth media with exosome depleted media. They do a couple of centrifugations prior to the main ultracentrifugation step at 100 G for 100,000 G for 1 hour, and then they purify it through OptiPrep, and then after that they collect the exosomes. They wash them a couple of times in PBS. And then they store them in -80 for a week. For the NKEMs, your starting point is the cells. So for most of the experiments, they use the same cells that they use to prepare the NK exosomes. So your starting point is the cells, and then they take the cells and they extrude them 8 times, each through 5 and 1 micrometer polycarbonate membrane filters using a mini extruder. And then after that, they had a final volume of 1 ml that they layered on OptiPrep, both a 10 and a 60%. And then they find the NKEM is enriched in the interface between the 10 and the 60. And then they take that and they ultracentrifuge it for 1 hour, and then they filter. And they mentioned that they store this also in the -80 for 1 month. There isn't more details in the paper on the stability, but from what they describe, they are more stable than exosomes. Next, please. So this is the figure. So panel A just explains the preparation method. Panel B is looking at the preps that they prepare. So comparing the NK cells with the exosomes, the NK exo and the NKEM. They loaded 50 micrograms of total protein. What you can see here, they're showing like tetraspin and CD63, Alex. You can notice right away that thank you. You can notice right away that you have that you can find in the NKEM. You can find sort of cytochrome C and GM130, which are proteins that you wouldn't regularly find in EVs that are like pertinent to the mitochondria or in protect them. And this is just an indication that these nanovesicles, of course, have different protein cargo than what you would expect in the exosomes. In panel C and D, they are looking here at the protein amount that they got from the same number of cells and the number of particles. And you can see, of course, that the EMs or the CDNs from the NK cells produced much higher amounts. In the paper, they say the particle number is 50-fold more than the exosome prep. Next, please. Thank you. Here, in this figure, they just show us the TEM and the nanocyte data, just for further confirmation that they have the right size and structure, morphology, and size distribution. So the next slide, I'll start describing the first assay that they did. So it was a cancer cell cytotoxicity assay. They used several cell lines in this work, but in the main figures, they only report the human glioblastoma, D54. So what they do with the cancer cell lines of interest is that they transfect them with recombinant retrovirus plasmids to express both efflux and THY1.1. So this makes them fluorescents, and then all the fluorescent cell lines have the same designation in a /F to indicate their fluorescence. So the generated stable cell lines were used for their assays, where they're able to do a readout by bioluminescence imaging, which they refer to in short as BLI. So the first assay I'm going to show you now, they incubated different concentrations of the EMs, the nanovesicles or the exosomes with the cells. And this is figure 3 here. So you can see in panel A, where they incubate either the control or the 3 concentrations of either the exosomes or the mimetics. And they show us the results for 24 hours, 48 hours, and 72 hours. And then you can see on in panel B, C, and D, the quantification of the bioluminescence in these images. And you can just see that, of course, the 30 microgram seems to have the highest effect, and it seems to be more pronounced with mimetics than the exosomes. In the next slide, we have, they look at NKEM or the mimetics biodistribution. So they give a systemic injection of these EMs. So they give a systemic, first, of course, they inject the cells and then subcutaneously into nude mice. And then 4 weeks later, the mice either receive IV injection of DID labeled mimetics at 100 microgram or PBS. Then they perform in vivo fluorescence and ex vivo fluorescent imaging to follow the biodistribution. So for the next slide, this is the results that they have. So you can see in panel A, they show us the localization by imaging here. So you find the PBS tumor, and then the 2 conditions of mimetics with either the mice have a tumor or don't have a tumor. And then you can see that in the case of the tumor, the mimetics are going to the tumor or colocalizing with the tumor. And then in panel B, they show us the ex vivo imaging of the organelles after harvesting, where you can see also where the mimetics distribute. And just notice that you can see, of course, in the middle panel of B, that the tumor has also indication of the EMs, like in panel A. And then in panel C, they quantify the various organs. And so what they conclude is that the mice with tumors had significant less uptake of the mimetics in the liver and the spleen compared to the mice without tumor. So in the next slide, they look at the in vivo cytotoxicity of the mimetics in the glioblastoma xenograft. So 10 days post xenograft, the mice are divided into 3 groups, each with 5. And then every 72 hours, these mice in either of the groups will either receive mimetics at a dose of 100 that is delivered intravenously, or at a dose of 30 micrograms that is delivered intratumorally, or PBS. And then they evaluate the signal intensity by BLI, and they also harvest the tissue. So the next slide shows a scheme of what I just described. So you can see that starting at day 0, 3, and 6, they either inject intratumorally or intravenously, and then every 2 days they will take an image that we'll see in the next figure. And after that, on day 14, there's the final images with the organ images. So in the next slide, we can see here the imaging that occurred every 2 days. And you can appreciate that for like at day 14, in the condition of the mimetics that are given intratumorally, that shows the least luminescence in that case. You can also see at day 14 in the control mouse that there's actually metastasis detected in the lungs. Other than that, pretty much the EM, the IT seems to have a stronger effect than the intravenous. If you look at panel B, it just shows the quantification of the luminescence in the images. So in the next slide, this is ex vivo imaging of the organs that were harvested. And in panel D, it shows also a graphical representation of that. And it confirms the same that we've seen with the actual whole mouse. And they say that they report that the control group exhibited 4-fold increase in signal intensity compared with the EM group. So in the next slide, they take ultrasound images of the tumors to further estimate tumor volume and tumor weight, and confirm the results that they got from the BLI imaging. And it also confirms the same that we have reduced tumor volume. Most reduced tumor volume you get is with the intratumoral mimetic injection. So in the next slide, they look at tumor cell uptake of NK mimetics. So what they did is they treated the mimetics with by labeling with DID, and they incubated it with either the mimetics or the exosomes for 3 hours. And then they did confocal imaging, where they did confocal imaging to see the uptake. And you can see that they were able to detect the uptake of the mimetics and the exosomes into the cancer cells, showing a similar interaction. And in the next slide, they started looking more into mechanistic aspects of this. As we know, NK cells exert their cytotoxicity activity through release of cytotoxic effectors contained in the Leydig granules, and also through transmembrane proteins like FASL and TRAIL. So they wanted to confirm, of course, the presence of FASL and perforin in the mimetics, and they did that by Western blot. And to further confirm the contribution of FASL to the cytotoxicity effect, they also utilized an inhibitor. And we'll see in the next slide, they report here in panel A, the Western blot data that they got for exploring FASL and perforin. So they looked at both the membrane component of FASL and the soluble cytoplasmic form of it, where you can see the presence of FASL and exosomes in the mimetics. They also confirmed the presence of the perforin. If we look at panel C, this is the experiment where they confirm the activity or the importance of FASL in this activity, where they show here that the mimetics versus the mimetic plus AF016, which is an inhibitor of FASL. So you can see in the presence of the inhibitor, in the case of the mimetics, you have the growth is almost as pronounced as the control. You can see a less effect in the exosomes. So although the inhibitor also increases the growth, but it seems to be less than the effect that it has on the mimetics. The other panel, the left side of panel C, is a result also of a proliferation assay. So they did the same setup as this plate, a corresponding proliferation assay to confirm these results. And in D also, they show the results for this assay. So in our next slide, they look into further into how the mimetics are affecting key proteins. So they're interested in proteins related to apoptosis and cell proliferation. So preferrin and granzyme, a lead, of course, the intrinsic apoptotic pathway that involves cytochrome C, while death receptor mediated apoptosis through transmembrane proteins like FASL result in activation of caspase 8, 3, and poly-ADP ribose polymerase and cell death. So they investigated these key proteins for apoptosis after 24-hour incubation of these cells with mimetics. And they also examined some of the signaling pathways related to cell proliferation, specifically MAP kinase and PI kinase, PI3 kinase. So in the next slide here, this just shows an outline of what they were looking at. So the proteins with a red asterisk are the ones that they followed up on with Western blot to indicate how they are affected with the presence of the incubation of the cells with the mimetics. So in the next slide, you'll see here in panel C, they investigate these apoptosis-related proteins, caspase 3, cleaved caspase 3, cleaved PARP, cytochrome C, and you can see that in the presence of the mimetics after incubating with the cells for 24 hours, that there's existence for these proteins. In panel D, they show signaling pathways related to cell proliferation. So we can see a reduction in p-ERK and phosphor-ERK, indicating less proliferation through these pathways. So in the next slide, I just summarize the finding that they report in this article. From the data that they presented, we can see that EMs or exosomes have similar size and morphology, but different in protein content. They report that the mimetics produced 50-fold more particles than what the same number of cells did produce exosomes. They confirmed by in vivo and ex vivo imaging the biodistribution, tumor targeting ability, and tumoricidal effect of the mimetics. And they suggest mimetics to be a potential alternative to exosomes, to NK exosomes for clinical applicability, demonstrating cost effectiveness and higher production rates. So in my next slide, I just want to bring to your attention another article that I found very interesting. I'm not presenting it, but I just wanted to refer you to it if you have more questions about this, of course. So the next slide. This is a really interesting paper that came out of the Hill Lab in Australia. So it's titled Proteomic and Post-Translational Modification Profiling of Exomimetic Nanovesicles Compared to Exosomes. And what this paper did is it systematically looked at using a specific human cell line. It was able to follow the differences between the cells and the exosomes and the mimetics. So I refer you guys to look at it for all the questions that you might have about different cargo. So in the next slide here, I'm just showing two figures of this paper, just to get an idea of what of how detailed this paper is. So in panel A, they just showed the difference on SDS page gel of the cell exosomes and mimetics. Panel B, they show you different markers. So they do mention in the paper that TSG101 is less prominent in the nanovesicle than it is in exosomes. And then you have a protein like BCL2 that's not typically found in exosomes. You can find it in the vesicles, in the nanovesicles. And then panel C, of course, which was the most important for me, is how diverse the proteins are between the various compartments, whether exosomes or cells or nanovesicles. And then panel D, it just shows the difference in PCI. some kind of analysis which just shows that the cells, that the nanovesicles are closer to cells than they are to the exosomes. And then in the next slide, there's this gene ontology figure, also an enrichment annotation that shows us the different category molecular function and biological processes between like the nanovesicles and the exosomes. This slide really shows how there's differences in what type of proteins are enriched in the nanovesicles versus the exosomes. And it just leaves us with a question of how much can we actually genetically engineer these cells to make nanovesicles closer to what we want in order to design our therapeutics or our particle of interest. So this is my last slide. My next slide is just thank you. I would love to take any questions. I'm really sorry. I wish this had been better, but I'm glad that Dr. Whitwer was available to save the day. And thank you so much for transitioning through the slides for me, Dr. Whitwer.
Sure thing, and thank you for presenting. We've had numerous questions in the chat box, and so I think I think that we see some common themes too in these questions. And from our conversations, you were wondering, what happens when you submit a paper with these, with mimetics to a journal? How are the reviewers going to respond to that? And it is quite interesting, I think, especially when you showed that figure from the Hill paper where they look at the relationship between the cells, the mimetics, and the EVs, that they see that there's a very, very close similarity with the total cellular compartment, but not so much with the EVs. And so I guess that does raise the question of why, what do we call these things to be accurate? If we call them an exosome mimetic nanovesicle or an exosome mimetic, maybe that would be something that would be made by extruding endosomes, that purified endosomes, or sort of something along those lines. And whereas with these other ones, the topology is different, the contents are different, and I think we're going to have some good questions here. But let me just start with a question that we had from Yvonne, and Yvonne's microphone is not working, so I'm just going to read this. She says, "I'd be keen to know your, Sarah's, anyone's opinions on using proteins as a way of measuring EVs for injection, especially in a situation like this." And you showed, I believe, Sarah, the figure that of the total protein content, and we could even look at then the ratios between protein and particle count. So Yvonne continues, "We know that different stimulation paradigms result in a shift in things like ceramide, and that this can disrupt the lipid protein ratios. I'm just wondering how much that's taken into account or how much it makes any difference." So this is about normalization of the input. So do you have any comments on that, Sarah?
That's a really great question, and I think it's a very critical aspect when you're trying to set up any of your experiments. What I think the best approach, regardless of course, the paper, because I guess the paper was depending on the concept of that they did as much purification as they could, and they just tried to unify across their experiments. But if your system is different, of course, or if you're putting new regulations to what you would do, I usually like to look at particle per microgram, and I like to try to modify as much as I can to get to the prep that gives me the highest value for that. And then I also, and it pretty much confirms nicely with Western blots. So when I've tried like various preps that I have that had like less particle per microgram versus ones with higher, I usually get a much better Western blot with particle per microgram. So estimation. So I guess it's really critical to make sure that your particles are pure enough before you try to draw any conclusions, because the more pure your particles are, you kind of set up the experiment for a better outcome, just because you're actually sure that you're putting in more exosomes than you are contaminants.
Good. All right. Thanks for answering that. So everyone, I'm going to try an experiment here. I'm going to go out on a limb and let people unmute themselves this time, because I'm thinking that we haven't had any problems in recent times So I'm going to, I'm just going to call on people. I'm going to go through the chat box, and if you can, just unmute yourself and ask the question. So the next question is from Maria Elena Ciccardi. Maria Elena, are you on? Are you able to unmute yourself and ask your question? Thanks.
Yes. So I was wondering, like, when they show the localization of this NKEM into the various organs, if they also look in the central nervous system. And I was also curious, like, what is the uptake mechanism, if they propose anything or if you know anything about it? The uptake mechanism for these particles.
Yeah, so the first part of your question, they didn't talk at all about CNS. And the second part of your question, if they suggested an uptake method, they don't. But all they refer to is that the reason they're getting uptaken is because of their similarity with exosomes. So it's just they're just assuming that they're being uptake for the same reason that exosomes are being uptake, just the molecules that are on the surface. And then there was another paper, like in 2013. It was a American Chemical Society paper that was where they actually trypsinized the vesicles or these mimetics, and then they weren't uptaken and no effect was happening. So it has to do with the proteins that are on the surface, and it probably is the same mechanism that exosomes get uptaken, but they don't talk about it in the paper, no.
Yeah, and so as far as the CNS, it looked like they didn't even show the heads of these mice, did they?
No, I don't think any of the organs were
unless
like,
you had cropped it. Yeah. Okay. Yeah.
No, so it was like liver, kidney, spleen, and tumor. I don't think there was CNS.
Yeah. So, so our next, so we also have a line of questions here about the procedures. So, Brian Bird, would you like to ask your question? I know you asked several questions, but the one was about the methods to make the mimetics.
Thank you. Yeah, I think that's really the only question I have is there any specificity when you put the cells through an extruder? Is this resulting in fragments of cells that are fundamentally different than if you just sheared the cells, tore it to bits in some other manner? Has anybody investigated that, this group or others, to see whether or not it's just any undifferentiated cell bits that do this, basically?
From the literature that I looked through, I didn't see anyone that did that specific investigation. There was also a review that I read that mentioned that you could also destroy these particles by sonication, ultrasonication, but nothing was talking about that specific question. And the more I read about how to prepare nanovesicles, I would really like to see some kind of like modeling of what actually happens to a cell if you try to squeeze it through a membrane. Like, I'm trying to imagine like what happens to the nucleus, what happens to all the organelles. Do something, are some like clusters in the receptors on membranes going to be more likely to end up in a vesicle or not? Like, I really think that that would be a really nice thing to see, like some kind of 3D visualization of what would actually happen.
Yeah, that's a nice idea.
Then we had some questions about the control group. So Phil Askanase, did you want to ask about the controls and make your comment?
I feel, can you hear me?
Yes, yes, we hear you.
I mean, these things have nothing to do with exosomes. We know that exosomes have a proteome and a transcriptome, et cetera. It's quite different from the cell of origin. So this is just the cell of origin. And we've skipped over the whole 3 billion years of evolution that got us to exosomes, of how the cell chooses what to put in these things. So they're quite artificial. And why would the body bother to have exosomes if it could just do this? The control is just set up as, I think it's a tumor line of NK cells for the source of the exosomes. So I'd want to work on that a bit. I'd like to have natural NK cells instead of tumor NK cells, and activated NK cells, et cetera, et cetera.
Yeah.
Putting them through this machine is activating them. And over on the control, it's an apples and oranges comparison.
Yeah, I agree.
Yeah, I do too. I think that, you know, that's why I think that they, we or somebody needs to come up with a good term for these cell-derived particles. And so some people just call them cell-derived nanoparticles or nanovesicles, CDNs, which I think is a better reflection of what they actually are. So I mean, it might be that there are some therapeutic effects that can be achieved simply by presenting bits of the cell in these small packets. But you're right, Phil, that this is not this is not the endosomal packaging and release pathway that we refer to as exosomes. This is something very different and much more cellular, as I think also Andy Hill's lab showed. So so next, I think that this is also part of the comments that several of our contributors had. So Tom Dredunks, do you want to continue with this theme?
Yeah, for sure. I'm happy I'm not the only one who is a bit skeptic of this paper. So I thought of another control. I wondered whether the cytotoxic effects that they write up here are due because they used NK cells passed them through an extruder, or could any other cell-derived nanovesicles have the same effect? And also, as a cautionary note, I was wondering if, because you do this, maybe some cellular proteins end up on the wrong side of the membrane. Could this cause autoimmune responses, for instance, or trigger inflammation of any kind?
Yeah, those are excellent points.
Yeah, so you're presenting danger signals really here. When presumably the nuclei are there, you're getting a lot of DNA probably in these preps. So there's a lot of things that are happening, potentially happening, that could be confusing the effects. So I think that somebody asked the question of also, what other controls do they need? They kind of will need to have a control with some other cell derived nanoparticles, nanovesicles. And then to assess, so I think that some groups have tried to do, they've tried to take nuclei out of the equation before they do the extrusions or the whatever cellular processing, and that might that might be something that can help. But then of course that's a very complicated process too, and it's unlikely to be 100% successful. So our next question is from Catherine. Catherine, do you want to ask your question about Figure 4? Hi, is Catherine here? I don't hear her. Perhaps she's having a problem with the microphone, but I'll ask this question. So this was about Figure 4C and the significance. How was significance assessed for the tumor when it was a comparison against a control group without a tumor? Do you, let me see if I can, maybe I need to pull that up.
Yeah, I can't actually see. It's just like a black. It's not like they actually don't have it. They just have it an empty spot. It's slide number 17. That's very interesting. Yeah, they just leave the spot empty.
And then the other part of this comment was that the total signal seems a lot less in the tumor-bearing mice. Total signal. Do you agree?
Yeah. So just looking at panel A. Just looking at how fluorescent the mice are. Yeah, that I agree, yeah. There's some background, maybe some background imaging issues there to address. Next question from Lilia. Lilia, would you want to ask your question about viability?
Yeah, thank you. I think actually my question is coming together with all of the previous questions where we're talking about the vesicles or not, and I'm always missing the part where they actually describe the cell viability and the apoptosis status and so on. Of the cells were like for the EV mimetic and so on. Maybe you showed actually one picture with the fluorescence where they kind of trying to prove that they're alive, or it's not about the cells of the region, or it's for the targeted one. And you know what I mean? Maybe that was
So are you referring to the one where they just looked for biodistribution?
Yeah, well, where they made actually those EV mimetic whatever particles, and they should have kind of the control of the cells from where they made them.
Oh, so maybe you're referring to
I haven't
the.
seen that. Mm.
Okay. Yeah, so maybe you're referring to the Western blot then, the first figure that was talking about the proteins.
Yeah, could be as well.
Okay. Yeah, so they don't really, I think something with these is that you really, anything can be in these vesicles. So I don't really know how you would do any type of quality control for them at all. And I don't know how you would even compare them. Like, I don't even think you can compare them with liposomes. I don't think you can compare them with EVs. I just think they're at its own. And I don't know how much regulation something like this would actually need. Because it's like exosomes are natural, so they're made for a reason, but you really don't know what are in these particles at all.
Okay. All right. Thank you.
Yeah, and you know, I mean, there's just a lot, I think, on this theme. So Kanda Reddy, Kanda, do you want to ask about the batch, the possible batches? Or maybe you don't have a microphone. In any case, what we see here in the chat box is a question about whether if you do this 10 times, you get exactly the same composition every time, or if, you know, as Phil suggests, maybe the cells are a little bit different, they're manipulated a little differently, and you get a completely different, you know, activation profile. And then DC in the chat box is also asking about the membrane profiles. As does Brian, you know, so these are some very serious questions. What is the topology of these membranes, and does the topology depend to some extent on the exact method of extrusion and how reproducible this is, how consistent this is from one batch to another. So I think that these are questions that are quite important, because they are going to also determine how the EV mimetic, or whatever you want to call it, the nanoparticle, is interacting with the recipient cell. You know, what it's displaying on the surface, and whether that's something that the cell expects to see, it's in the right topology, or if it's something that would normally be inside. I then wanted to I want to give the mic over to Brandon. Brandon, you had a nice question about a completely different method. It's not a physical method, but something else that could be used for making nanovesicles. Can you tell us about that? Hi, is Brandon here? Brandon is Brandon is asking about CDN, so cell-derived nanovesicles that could be made by chemical induction means. So here, I guess you're so as an example, self-hydro blocking. So if you do this, if you treat cells this way, maybe then I mean, this is I guess a completely different method for making a lot of vesicles from a cell. Sarah, do you have any experience with that, or have you seen anything on chemical induction in the literature?
No, I haven't, but that's interesting.
And this might be a way to you know, to really use membranes in the topology that they're supposed to be, without some of that deep cellular contamination. Erez, you're up next.
So first of all, I want to say that I completely agree with Philip and probably the rest of the people here that those are not exosomes, and exosomes mimetic are a very, very bad name for them. I do think that there is a therapeutic potential for this technique, and like I just wrote, from a batch-to-batch perspective, it's probably similar to exosomes. It's also changing based on the cell condition and so on. So I think that in that regard, there is obviously a challenge there, but it doesn't say that you cannot streamline that into something that has therapeutic potential, at least in my mind. For me, it was missing the effect on the immune system, because as you say, there is a danger signal on those exosomes. There are many more, or not exosomes, sorry, on those cell particles, my bad. And you would expect them to do much more than just do fast ligand signal cell lysis, but really affecting the mice systemically and the immune system systemically.
Yes, I agree.
Yep, a lot of different effects that we might have to look for with these sorts of materials that we wouldn't necessarily do with naturally produced EVs. All right, so one final question is from Maria Kuipers. Maria, do you want to ask about the densities?
Yes, so thank you for the presentation and interesting discussion following. So I was wondering, because I looked through the materials and methods section, and I saw that they mentioned that they build a iodexonal gradient with 2 ml of 60% and 2 ml of 10%. Then they loaded the sample on top, and then they took the what is it? The interface between these two gradients. And I was like, yeah, that could be 2 ml, 4 ml, it could be 30%, it could be 20%. It doesn't say anything exactly about what kind of fraction they took these NKEM from. So I was wondering, because they referred to 2 papers where they got this protocol from, whether they, in these papers, they have mentioned these exact fractions.
I don't remember. I just remember in the last paper I showed you, the proteomics one, that one had like, so yeah, there is one. I just can't, like, I read so much papers, I can't remember what I saw it in, but there was a paper that collected 7 fractions, and they ended up finding it in the fraction number 5, but I just can't remember which paper it was. It wasn't this one. It could be one of the references paper that they had, but I just wish I could remember, but I don't remember which. But some papers did do that. This paper didn't. This group had previous work with nanovesicles. They had like a paper about melanoma. So maybe maybe that's where I read it, but I just not 100% sure. But I did see papers that did fractionation, and then they decided according to, I think they made the decision according to nanosite, if I'm correct, about which one to pick. Yeah.
Okay.
Yeah, so certainly a very good point that if we want this to be reproducible, we have to know exactly what those fractions are, and how to make them. So, well, that brings us to the end of our hour and the end of this journal club. So thank you again, Sara, for being so generous as to share your time and your expertise with us. And I think we've learned some lessons from this paper, and maybe raised some questions about exactly what EV mimetics are. So thank you, everyone. I hope that you all have a great rest of the week. Stay safe and healthy, and we'll look forward to seeing you again next week. Bye now.
Bye.